Okay, so good evening, everyone. So welcome to the week 3 problem solving session of this NPTEL course, Genetic Engineering. So let's wait for 2 to 3 minutes and then I'll start today's session. So I hope all of you have gone through the week 3 lectures. So in week 3, what all topics were covered. So in week 3, we are starting to learn the basics of cloning. So in genetic engineering, one of the main concept uh, or one of the main theory is gene cloning. So what are the various ways why is gene cloning required, and how we can clone genes in a vector and then transform it in a host cell, and then we can screen whether our clone gene is present in the vector. So all these, so what are the different techniques that are used? So, so these are the concepts that were covered in week 3. So today we will just quickly brush up all the concepts that were covered in week 3, and we will also solve a few sample questions so that through the questions you can understand the concepts even better. Um, so starting from what is gene cloning, and then when do we need to clone a gene, and what are the ways by which we can clone a gene, when to go for genomic library screening or cDNA library screening, and then finally how to screen these different clones, and finally if a particular gene is already known or the sequence of a gene is known, then we can directly go for cloning using PCR amplification, isn't it? So we PCR amplify a particular gene sequence, and then the amplified product is digested with restriction enzyme. The vector is also digested with the same restriction enzymes, and then ligation is done. So ligation reaction is set up between the to ligate my vector with the amplified gene product, and then following that, what is the transformation? So we transform that ligated gene product into our host cell. Mainly, commonly or most widely used is the used host cell for most of the applications are E. coli. Okay. So, okay, so I'll start today's session now. Okay, so in case in between the lecture, if my slides are not visible, or if if you are facing any issue with the video, or my audio is not visible, then you can at least write it in the chat and let me know, because last week I saw that the later half of my video does not have any slide. So somehow the slides were not being presented. So please let me know. Okay. Yeah. So good evening, everyone. I am Akash Jyoti Pathak, and I'm one of the PMRFTAs for this NPTEL course, Genetic Engineering, Theory and Application. This course is being run by Professor Vishal Trivedi of IIT Guwahati. So, so a quick recap of what all topics were covered in week 3. So I have been yeah. So the topics that we learned in week 3 were the basics of cloning. So there are basically two parts of this cloning. So in week 3 also, some part of the cloning has been covered, and in week 4, uh, again we will go into further details. We will learn further details of the cloning. What are the different vectors that must be used, and other details we will see in week 4. So in week 3, we learned about when is cloning required, right? So so cloning will be required in two cases, first of all, right? So if the target gene sequence is known, so if the target gene sequence is known, if it is known, then what we can do, so if the target gene sequence is known, then we can directly go for PCR amplification of that particular gene sequence. So we can design the forward primer and reverse primer. So for designing the forward primer and reverse primer, first of all, what is required? We need to know the gene sequence. If we do not know that gene that is to be cloned in our vector, then we cannot go for PCR amplification. So for PCR amplification of a particular gene sequence, or the sequence must be known. Then only we can clone it using PCR amplification, isn't it? Okay. But if the target genome sequence is not known, then we can go for two methods. So either it is genomic screening or it is cDNA library screening. Okay, genome DNA library screening, or it is cDNA library. So we have to screen for the So we have to screen for the target gene in the library, in the genomic library or in the cDNA library, depending on what is our purpose. So if like for human genome sequencing, when it was not known that which part of the human genome is coding, which is non-coding, how many genes are present. So for such cases, what is done? The genomic library is prepared, right? And then after the genomic library is prepared, then we can screen that which particular region of the gene is coding for mRNA, which is coding for tRNA, or which part is non-coding. So such screening is then done, and then we can annotate the regions of each region, the regions of the genome, that which part of the genome has which function, has what function. Okay, or it can be generation of cDNA library, and then what is done? Then screening of recombinant colonies and polymerase chain reaction. Or so we will look into these concepts in detail. So let's go one by one. Okay. So yeah, this is directly from your lecture slide. So first of all, so if the gene sequence is not known, then we can screen the genomic library or the cDNA library. So when do we need to screen for the cDNA library? So the cDNA library screening is required when we only want to know the part of the genome which is coding for a particular protein, isn't it? Because the cDNA, what will it have? It will not have any introns. It will only have that part of the genome which uh, which will be first transcribed into mRNA and then translated into protein. So that is why it is transcriptome. Transcriptome means only that part of the genome which will code for a protein. Okay, and then screen for it. So you clone the different fragments. So we will see how in the subsequent slide, we will see how a genomic library is prepared and how a cDNA library is prepared. And then how do we screen for a particular gene in these libraries? Okay, and if the gene sequence is known, then directly we go for polymerase chain reaction. So in the polymerase chain reaction, uh, by polymerase chain reaction, what we will get? We will get a fragment of gene that will be amplified, and then this amplified DNA fragment is cloned into a suitable vector. Okay. Okay, so what is gene cloning first of all? So this figure is taken from the book T.A. Brown. So you all know you have studied gene cloning in class 11th and 12th, isn't it? So basically what we do, so we know, so if you see this figure over here, so you have a fragment of DNA and you want to insert this fragment of DNA into a vector. Okay, so this is your vector. So what you do, basically you from the genome, you cut a fragment of DNA using restriction enzyme, and then you cut the vector with the same restriction enzyme. So we will, in the next lecture, we will see what are the different restriction enzyme and all these concepts we will see. But here we are starting, we will learn about the basics of cloning first. So then what is done? So ligation is done. So on ligation, what will happen? The vector will be uh, or the fragment of DNA will be cloned inside the vector. And then once you get, so this is called the recombinant DNA molecule. And then this recombinant DNA molecule it is transferred into a suitable host. So here in this figure, it is shown that the host cell is bacterium. So in our back inside in the bacterium, so in the second step, what is happening? The recombinant DNA molecule is getting transported into the host cell. So you see here, so the bacterial cell was empty. Now, after transformation, this bacterial cell is containing the recombinant DNA plasmid. And then this recombinant DNA plasmid will be copied multiple times. So what are the different um, regions that a plasmid must have? That also we will see in the next week. So it must have OD site and selectable markers and start site. All these things we will see in the next lecture. So yeah, so it will get multiplied. So multiplication of the recombinant DNA molecule and division of the host cell. And then in the next stage, what is done? After numerous cell division, we will, so when this, after transformation, when this culture is plated on a LB agar, we will see colonies. We can find colonies that have been formed on these LB agar plates. So what is gene cloning? So basically a fragment of DNA containing the gene to be cloned is inserted into a circular DNA molecule called a vector to produce a recombinant DNA molecule. So this is your fragment of DNA. This is containing your gene of interest, and this gene of interest has to be cloned into a vector. So vector is again a circular DNA molecule. So what is done? So the vector, why is called a vector? Because vector is helping in the transport of the gene into a host cell, which is usually a bacterium, although there can be other types of living cell as well. So we can use mammalian cell as well, but in that case, it is called transfection. So within the host cell, the vector will multiply and produce numerous identical copies, not only of itself, but also the gene that it carries. Okay, so because your vector, your recombinant plasmid is also containing your gene of interest, once is containing your gene of interest along with the multiplication of your vector, the gene of interest will also get multiplied. So when the host cell will divide, the copies of recombinant DNA are passed to the progeny. So yeah, so binary fission is taking place, and along with the division of the bacteria, the plasmid is also getting transferred into the progeny or passed to the progeny. And after a large number of cell divisions, a colony or a clone of identical host cell is produced. So each cell in the clone will contain one or more copies of the recombinant DNA molecule. And the gene carried by the recombinant molecule is now said to be cloned. So this is what is gene cloning. Okay. Now, now what is the problem of gene selection? So for example, so first I will cover this slide first. So first of all, when is gene selection required? So when you have a genomic library, so in the genomic library, what is done? First of all, you have a cell. Okay, you take any cell, and it can be mammalian cell, it can be any bacterial cell. So depending on whose genome you want to study, so there can be different genomic libraries. So first of all, what is done? The cell is lysed. The cell is lysed, and once the cell is lysed, you will have the nuclei. Now from the nuclei, you get the DNA or the chromatin, and now this chromatin is fragmented. Chromatin is fragmented by treating it with a particular restriction enzyme. Okay, so there are multiple restriction enzymes that are available. So it can be either any common restriction enzyme you can use, like EcoR1 or SAU3A or BAMH1. And these restriction enzymes, what will it do? It will create DNA fragments. So it will create small DNA fragments so that your entire DNA will get broken into different into small DNA fragments. Now then this DNA fragment is inserted into a suitable vector. Okay. Your vector, the vector will also be treated with the same restriction enzyme, and then these different DNA fragments will be ligated with the vector and transformed into a host cell. Now, because there will be different types of recombinant plasmid that will be produced, you have to screen for the your gene of interest. So, which recombinant plasmid is containing your desired gene of interest, that you will have to screen. Okay, so that is why there is this problem of gene selection. So the problem is to obtain a clone of a single specified gene. Okay. So here if you see this figure here, so uh, so 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 here in this figure, what has been shown is that you have a 50 kb DNA molecule, 50 kilobase per DNA molecule, and then this DNA molecule is digested with EcoR1. So once you digest it with EcoR1, you will get multiple DNA fragments. Okay, some will be of longer, short fragments, some will be long fragments, and out of all these fragments, this is the particular DNA fragment which in which you are interested. Okay, so you get many DNA fragments, and then all these DNA fragments are cloned into into a vector, into the vector. So you see here, so this fragment is cloned into one of the vector. This fragment that you get, this is cloned into one of the vector. See, all these fragments have been cloned in different in the vectors. Now you want this your interest, but your gene of interest is present in this particular vector. So that is so out of all these different recombinant plasmid, you have to screen that which of the colony that you are getting has the plasmid with your gene of interest. Okay, so that is what is mentioned here. The problem is to obtain a clone of a single specific gene. Okay, simple organisms like E. coli contain several thousand genes, and the restriction digest of total cell DNA produces not only the fragments carrying the desired gene, but also many other fragments carrying all other genes. Okay, so the fragments you get, that will have not only have your desired gene of interest, but it will have multiple genes, different types of genes will be present. Okay, so because your genome will have, or you have the bacterial genome, if you see the bacterial genome itself has thousands of different genes. Okay, so now during the ligation reaction, there is no selection of for an individual fragment. Okay, so during when you are doing performing the ligation reaction, then your vector is getting ligated with any one of these vectors. So there is no selection during this process. So And and what you are getting different recombinant DNA molecules. So all of which contain different pieces of DNA. Isn't it? So a variety of recombinant clones are obtained after transformation and plating. Yeah, so what you have to do, you have to find the correct one. The correct one must be identified. So we will look into how this screening is done. So now how is genomic library prepared? So same thing. So you isolate the DNA, the whole genome or the DNA from the cell, and then you treat it with restriction enzymes that will give you several different fragments, and then you ligate it into a vector and then transform it into a host, this recombinant plasmid. So a genomic library is a set of recombinant clones that contains all of the DNA present in an individual organism. So it is because it is genomic library, it will have all the DNA. The entire DNA of the organism will be cloned in the will be present in these recombinant vectors. Okay, so an E. coli genomic library, for example, contains all the E. coli genes. So any desired gene can be withdrawn from the library and studied. Now, genomic libraries are prepared by purifying total cell DNA. So first you have to purify the total cell DNA, and then what is done? A partial restriction digestion is done, and this will result in fragments that can be cloned into suitable vector. So usually a lambda replacement vector, a cosmid vector, or it can be a yeast artificial chromosome. So depending on from which organism you have isolated the genome, whether you have isolated the genome from a human cell or whether you have isolated from a bacterial cell or a plant cell, depending on that you have to choose a particular vector. So if it is a human genome that you have isolated, then the vector, suitable vector, or you cannot use bacterial plasmid vector, because in that case the if you fragment the human genome, then you will get large fragments, large DNA fragments, and the each vector. So there are different types of vectors, like there can be plasmid vector or phagmid vector or cosmid vector. So each type of vector has can uh, carry a particular number of DNA base pairs. So depending on what is your genome, or what is the genome uh, you have or what is the organism from whose genome you have isolated, yeah, accordingly you have to select the particular vector. So this type of library is most useful when generating DNA for sequencing a genome. Okay. So for example, when a human genome sequencing was done, the human genome was genomic library was prepared by cloning these fragments in yeast artificial chromosome vector. Okay. So it can these type of vectors can carry large size insert or inserts which are having more number of DNA base pairs. Okay, so for an organism with more complex genome, this type of library is not suitable for this task, because many of the DNA inserts will not contain coding DNA sequences. Okay, so for example, if you want to do genome sequencing, then you need to prepare genome libraries. But if only you are interested in finding or finding that what are the genes that are getting expressed in a particular cell, or 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 you are concerned with the particular protein or expression of a particular protein, then there is no requirement of generation of genomic libraries. So in that case, what do we go for? We go for cDNA library generation. So first we will see how we construct a genomic library. So first of all, for construction of genomic library, what is done? The genomic DNA is isolated. Okay. So once the genomic DNA is isolated, because the genomic DNA is very large, it is fragmented by digestion with the suitable restriction enzyme. So here in this figure, you can see it has been digested with EcoR1. And once this human DNA is digested with EcoR1, you are getting several DNA fragments. Now, similarly, the vector. Your vector must also have this restriction enzyme site. So this is yeast artificial chromosome vector, because you want to clone the human DNA fragment into this the or prepare a genomic library of the human DNA. Therefore, what you have to, you are taking this YAC vector, and then this YAC vector is having this BAM-H1 site and EcoR1 site. So you have treated this vector with BAM-H1 and EcoR1. So once you treat with this, you do a double digestion of this vector, you get two fragments, DNA fragments, and then recombination or DNA ligation is done. So when this DNA ligation is done, what will happen? You can see here that this EcoR1 site and this EcoR1 site, they will join with each other because of the complementary DNA sequences, right? And you will get this recombinant DNA plasmid. Isn't it? And then once you have the recombinant DNA plasmid, you transform it in a host cell. Okay. Because this the DNA fragment that you are getting by digesting with EcoR1, the human that will have both that will have this EcoR1 sticky ends at both their ends. So the sticky end, one of this end can join with this EcoR1 sequence which is present in this fragment, and the other gets um, ligated to the other EcoR1 site which is present in the vector. Now, okay, so these are the four steps. So what are the four steps? First, you will isolate the gene, isolate the genome, then you will fragment it by treating it with restriction enzyme. And then you will, then what is done? Then ligation. Ligation of the DNA fragment with the vector, with a suitable vector, and the fourth will be transformation. So after transformation, what else needs to be done? Screening. So now what are the steps that are involved in isolation of genomic DNA? So yeah. So this was explained in the lecture as well. So you have a cultured cells or tissues. Now this cultured cell or tissue, what will be done? The cell has to be lysed first. So how will the cell be lysed? So you treat it with SDS. So the SDS, what will it do? It will disrupt the cell membrane. It will disrupt the cell membrane, and all the contents of the cell will you will get the contents of the cell. Now you do an enzymatic digestion. So the DNA will also be isolated. So you digest it with EcoR1 or any restriction enzyme, which is with whose restriction sequences are very common in the genome. And then after enzymatic digestion, we precipitate it with absolute alcohol. So after precipitation with absolute alcohol, the genome is purified with phenol chloroform treatment, right? And so basically, what is happening? You have the cell, you lyse it. Okay, you lyse it by treatment with SDS and protease K. So why do you add protease K? So basically, if you the DNA inside the cell is wrapped around by histone proteins, right? So you want to digest these histone proteins or that's DNA protein complexes. So the DNA is not free; it is complex with protein molecules. So you want to digest the protein as well, so that you can get the pure DNA. So you treat it with protease K. So this protease K will digest all the protein complexes with which this histone is which this DNA is associated, and now you will get the genomic DNA. So once you get the genomic DNA, you treat it with a particular restriction enzyme. So once you treat it with restriction enzyme, you will get different fragments of DNA. So now in your solution, you will have these different DNA fragments, and along with the different DNA fragments, you will also have these SDS and protease K and all these other components will be also present. So to separate these DNA fragments from these other components, from this SDS and protease K, and alcohol precipitation is done. So basically what you do, you add 100% ethanol. Okay, so 100% this solution is treated with three volume of 100% ethanol. So for example, you are having this has around, for example, 500 microliter of your sample. So you treat it with 3 times of 500 microliter and you precipitate the DNA fragments, and you spin it. And when You spin it, you get a pellet of the DNA fragment, and all other components remain in the supernatant. So the supernatant is discarded, and then what is done in the next step? In the next step, the pellet that you get, the pellet is resuspended with phenol chloroform. By phenol chloroform treatment, so once you treat this DNA fragments with phenol chloroform, then you get two different phases. One is an organic phase and another is an aqueous phase. So your aqueous phase has contains the DNA of interest, or has your DNA. Basically, the aqueous phase will have your DNA, and the organic phase will have all other protein and other components. So it will mainly have protein. And then the aqueous phase is isolated, and then again this aqueous phase is again precipitated with absolute alcohol, so that you can remove this phenol chloroform from your solution. Okay, and then what you are left with is a pure mammalian genomic DNA, which you can analyze on by running an agarose gel, 1% or 0.8 to 1% agarose gel. If you get a very clear, crisp single band, then you can be assured that yes, you have isolated the pure genome from your cell. Okay. So what are the steps? So the first step is you slice your cells with the detergent containing lysis buffer. So what will your lysis buffer have? So your lysis buffer will mainly contain SDS, and then the incubation of these cells with digestion buffer containing protease K SDS to release genomic DNA from DNA protein complexes. So your protease K, basically it will it will degrade all the protein which is present in the to which your DNA is bound with, and then you isolate the genomic DNA by absolute alcohol precipitation. Okay, so once you do the absolute alcohol precipitation, all the DNA fragments will get precipitated, and and you can then remove this SDS and protease from the solution, and then you purify the genomic DNA with phenol chloroform mixture. So you get an aqueous phase and an organic phase. The organic phase contains your protein, and your aqueous phase has the DNA. So the remaining, so even after protease K treatment, any other protein or remaining protein that are present, that will all get separated in the organic phase. And then the genomic DNA, which is present in the aqueous phase, is precipitated with absolute alcohol. And then you analyze the genomic DNA by running a 0.8% agarose gel. Okay. So answer this question now. So the enzyme that is commonly used for restriction digestion of genomic DNA is option A, RNase. So you can also answer it in the chat as well. Okay, so what are the enzymes? So RNase will, what will it do? RNase will digest the RNA. RNA is for digesting the RNA. Now, what you want to do, you want a restriction in enzyme. Okay, so also your option must have one of the restriction enzyme. DNA polymerase is used for DNA replication. Protease K is used for digestion of the protein complexes to which your DNA is bound. And so the only option is EcoR1. So any if your option has any one of the restriction enzyme, then that will be your answer. So EcoR1 is a commonly used restriction enzyme in molecular biology, and it is used for digestion of genomic DNA. This enzyme recognizes the specific DNA sequence GAATTC and cuts between the G and A on each strand, creating sticky ends. These sticky ends are single-stranded overhangs that can easily pair with complementary sequences. So for restriction digestion of genomic DNA, you require a restriction enzyme, isn't it? So the only restriction enzyme which is present in the options are or EcoR1. So option D is correct. Okay. So So now the vector that was used to create the genomic library for human genome sequencing project. So just now we discussed this. So for the human genome sequencing project, the genomic library that was used was yeast artificial chromosome, because yeast artificial chromosome can carry large DNA fragments. So you can clone large DNA fragments in yeast artificial chromosomal vector. You cannot use bacterial plasmid vector for cloning of human genome fragments, isn't it? Okay, so answer is yeast artificial chromosome. So here, so this table was there in your lecture slides as well. So your plasmid. So for cloning an insert in a plasmid vector, your insert can be 15, 15 megabases. For Phage lambda, it can be 25. Plasmids, it can be 45. For bacteriophages, 72. We can clone inserts having 70 to 100 base pairs, or and for bacterial artificial chromosome, 120 to 300 mega base pairs, and yeast artificial chromosome, you can use 250 to 2000 mega base pairs, okay, or million base pairs. So into 10 to the power 6, all of this is 10 to the power 6. So the yeast artificial chromosomes are crucial in the human genome project for creating a genomic library by cloning large DNA fragments. Okay, ranging from 100,000, that is 100,000 means 1 lakh to 1 million base pairs. So the human DNA was partially digested, inserted into yeast artificial chromosome vectors, and introduced into yeast cell. Now, after introduction into yeast cell, it could be replicated, and so the yeast artificial chromosome clones were screened, mapped to human chromosome, and used as templates for sequencing, enabling the assembly of human genome. So the yeast artificial chromosomes allow the handling of extensive genomic regions, facilitating the project's goal of sequencing the entire human genome. So because you see here that you can clone large fragments of DNA inside each artificial chromosome from starting from 250 to 2000 mega base pairs. So into 10 to the power 6. So that is why the for cloning fragments of human genome, each artificial chromosome vector was used. Okay. So the limitation of genomic DNA library is, so what are the options? They are difficult to prepare. They contain sequences with no information on gene expression status. They cannot be used for large genomes. They are expensive to maintain. So what will be the option? Correct option. So one of the main because what is the main limitation of genomic DNA library is that your genomic DNA library will contain both coding and non-coding regions of the DNA. So the entire DNA of the organism will be present in the genomic DNA library. Both coding and non-coding DNA sequences will be present. That is why it becomes difficult to so 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 if you are only interested in identifying or only the particular region of genome which codes for a protein, then this will be genomic. It is not possible to screen a genomic DNA library because there will be a large amount of sequences that you will have that has to be screened. Okay, that have to be screened. That is the one of the limitation. Okay, so that is what is there. So answer is option B actually. So a limitation of genomic DNA library is that they include all the DNA sequences, the entire DNA sequence from an organism's genome, regardless of whether those sequences are involved in gene expression. This means that the genomic library contains not only coding regions, but also non-coding regions such as introns, promoters, and intergenic regions. So as you know, in the molecular biology, if you have studied, you will know that these introns do not code for any protein. But even these will be DNA sequences will be present in your genomic library. The promoters, the intergenic regions, the regulatory genes, all these will be present in the genomic library. As a result, these libraries provide no direct information about which genes are actively expressed or the level of their expression. So to understand gene expression status, complementary DNA libraries are generated from mRNA and thus represent only the expressed genes. Okay, so we will learn about complementary DNA library. So I hope this question is clear. So because genomic, if you when you're screening a genomic DNA library, there will be lot of information or lot of DNA sequences that has to be screened. Because the genomic DNA library will not only have your coding regions of the genome, but also it will have the non-coding regions. So what are cDNA libraries? So So basically, if you know basic molecular biology, then what is the flow of information or the central dogma of molecular biology is that the information flows from DNA to mRNA and from mRNA to protein. So the information which is present in DNA is copied to mRNA or transcribed to mRNA, and then this information is transferred from mRNA to protein. Now, so basically, what will the mRNA contain? It will contain only the information that is necessary for synthesizing a protein. So if the mRNA, so basically if you consider an eukaryotic genome, then it will have what? It will have both introns and exons. So introns and exons. So the exons which are flying by the intron sequences. Now, the only the exon sequences code for the protein, while the intron regions do not code for any protein. They are non-coding regions. But your mRNA, what will it contain? It will only contain the exon regions of the gene. It will have only the exons. So if you can reverse transcribe the mRNA to DNA. So by using reverse, so the viruses have one type of enzyme called the reverse transcriptase enzyme, and using the reverse transcriptase enzyme, we can synthesize the complementary DNA from mRNA. Okay, so complementary DNA. What is complementary DNA? So when we are reverse or when we are synthesizing the DNA from mRNA, then that type of DNA is called the complementary DNA. So the complementary DNA does not contain any intron sequences. It will only have the exon sequences. It will only have those regions of the gene or that will code for a protein. Okay. So so here in this figure, so these the figures are taken from this T.A. Brown book. So you see, so this is the particular cell. So this is the genome which is present in cell type A, and the same genome is present in cell type B as well. Now, depending on the, as you all know that depending on the cell, a particular gene is expressed or a particular gene is repressed or it is not allowed to be expressed. Okay. So for example, if it is your lung cell, then a particular gene will be expressed, or for example, if your stomach cell is there, then it will express different type of enzymes for digesting your the food. But if it is a lung cell, then those type of genes will not be expressed. For example, if it is a pepsin gene or a pepsinogen gene, then that particular gene will be expressed only in the stomach cells, but that gene will not be expressed in our lung cells. So, but the genome that is present, that is same in both, in all the cells of our body. So all the cells of our body will have the same type of genome, will have the same genome, identical genome, but the expression of the genome will be different in different cells. So that is what is shown in this figure. So there are so in cell type A, these particular genes are silenced. So the ones that are marked in gray, those are silent genes, while those that are marked in red, they are the genes which are getting expressed in cell type A. While in cell type B, what is happening? Other types of genes are getting expressed, but the genome is same in both the cells. So now, if you want to understand that what type of gene is getting expressed in a particular cell, then you can do the screening of the cDNA library. So what is your cDNA? So to enrich for coding sequences in the library. So basically the cDNA contains only the coding sequences in the genome. That is why if you want to enrich the coding sequences in the library, a cDNA library is created. So instead of starting with the genomic DNA, mRNAs are converted into DNA sequences. So if you convert the mRNA to DNA by using the reverse transcriptase enzyme, then this type of DNA is called the cDNA. Okay, the process is called reverse transcription. So this process is called reverse transcription, and the enzyme that is used is called the reverse transcriptase. And this can make DNA from an RNA template. When treated with reverse transcriptase, mRNA sequences are converted into double-stranded DNA copies called cDNA. So that is so next what it is explained. So that is what I was explaining. In different cell types, so in different cell types, different sets of genes are switched on, while the others are silent. Only those genes that are being expressed are transcribed into messenger RNAs. Okay. So what is the meaning that a particular gene is getting expressed? It means that a particular gene will be transcribed into mRNA, and from that we will get protein. So only those genes are getting expressed. So if the mRNA is used as the starting material, then the resulting clones would comprise only a selection of the total number of genes in the cell. So you will get what are the particular genes which is present in the cell, because only that mRNA will be generated for whose gene is getting expressed. So you will have your cDNA will be only though will contain only those genes that are getting expressed in particular cell. Okay. So how do you generate a cDNA library? So you understood how a genomic DNA library is prepared. So first for the generation of genomic DNA library, you were lysing the cell. After the cell was lysed, then the DNA was fragmented by treating with restriction enzyme. Then it was precipitated with absolute alcohol, and then you treat it with phenol chloroform, where you get an organic phase and an aqueous phase, and from the aqueous phase, your aqueous phase will contain the genomic DNA, the entire genomic DNA. Okay, but for cDNA library, what is required? So the starting point will be isolation of mRNA. So if you have to generate a cDNA, then what will be required? The main starting material that will be required is isolation of mRNA. So the first step is isolation of mRNA. So your cell will, the nucleus of your cell will have different types of RNA, mRNA, rRNA, tRNA, right? So first you'll have to isolate the mRNA, and then from the mRNA, so once you isolate the mRNA, then inside this, what you can add reverse transcriptase enzyme. So this reverse transcriptase enzyme is commercially available. You can add this reverse transcriptase enzyme in your append of tube, and then from your mRNA you will get the complementary DNA. So it will the mRNA will get reverse transcribed to DNA, and you will get the complementary DNA. Once you get the complementary DNA, then what you can do, you can you can again do the cloning, follow the cloning method. So you treat your complementary DNA with a set of restriction enzyme, and you treat your vector with the same restriction enzyme. You clone or you ligate the complementary DNA with your vector, and then once you get your recombinant DNA plasmid, then you insert it into a host cell, and then this host cell is allowed to grow. So after you grow the host cell, then again what you do a mini prep, or you isolate the plasmid from the host, and then you sequence it. So once you sequence the gene, then only you will be assured that yes, uh, your clone that you have the clones contain your desired gene of interest, or it has your cDNA. Okay, these points are clear, isn't it? So So now there are various methods by which you can get a complementary generate or synthesize a complementary DNA. So, but in all these methods, the first starting point is that you need an mRNA. Okay, so first you will have to isolate mRNA. Now, one of the method is homopolymer tailing. So basically, if you lyse the nucleus, there will be you will get different types of RNA, right? mRNA, rRNA, tRNA. And now, if you know basics of molecular biology, then only mRNA has a poly A tail. So if you see the structure of an mRNA, so it has a methyl 7 methyl guanosine residues are present in the head group, and in the tail regions you have poly A tail. So poly A tail is present in the mRNA tail. So this poly A tail is only present in mRNA. So if you want to purify the mRNA from your RNA population, then what you can use an oligo-DT column. So what is an oligo-DT column? So you have a column, and in this column you have resin. So these resins are conjugated. So these resins, if you see these beads, so these are the beads, and these beads are conjugated to your oligo-DT column, which means that these will have thymine base pairs in that oligo-DT means that your DNA fragment, that this particular DNA fragment will have thymine base pairs. Okay, several thymine base pairs. So when you add this RNA into your column, then only what will happen? Only the mRNA which are having this because this mRNA will have this poly A tail, only they will bind to your column, only they will bind to these resins which are conjugated to oligo DT primers, and then while the other RNA fragments they will get washed off. Okay. So in this way, you can isolate the mRNA from the other RNA population. Okay. So once you get this mRNA, so once you have your pure mRNA, so from here you will have your pure mRNA. So once you have the pure mRNA, then what you can do, then you can do reverse transcription. So basically your mRNA will have this poly A tail, and you can take a, what you can take? You can take a oligo DT primer. So you take an oligo DT primer. So your primer will have this TTTT base pair sequences, and this will bind to this to your mRNA, and then once you add DNA polymerase, what will happen? It will extend the 3' OH group, which is the free 3' OH group, which is present in this thymine, and it will polymerase in the 5' to 3' direction. Okay, and you will get a new DNA strand. So this is your cDNA that you will get. Okay. Now there are different mechanisms by which you can methods by which you can so you get a RNA DNA hybrid, isn't it? So there are different mechanisms by which this RNA can be degraded, or you can remove this RNA, and you can be only get your DNA. So one is homopolymer tailing. So what is done in homopolymer? So what is homopolymer? Polymer in which all the subunits are safe. So DNA strand, for example, made up entirely of deoxyguanosine is a homopolymer and is referred to as polydeoxyguanosine or poly-DG. Now, tailing involves the use of enzyme terminal deoxynucleotidal transferase to add a series of nucleotide onto the 3' OH termini of a double-stranded DNA molecule. So if you have your DNA molecule, Okay, 5 prime to 3 prime, and your 3 prime will have a free OH group, isn't it? Now, if you want to a particular type of nucleotide to get attached to your DNA, then what you can use the terminal transferase enzyme. So what this terminal transferase enzyme does is that it adds a particular type of oligonucleotide to the 3 prime end of the DNA. So if you want to have a poly DG 3 prime tail, and then what will it do? So if you add this terminal transferase enzyme, and if you add DGTP um, to your reaction, and so what it will do, it will add the guanosine residues to the 3 prime end, and you will get a polydeoxyguanosine molecule. Okay. So telling to add a series of nucleotides onto the 3 prime OH, so we will have GGGG at its end. Okay, if this reaction is carried out in the presence of just one deoxyribonucleotide, a homopolymer tail is produced. So that is what I was telling. So if you want to add number of G residues at your 3 prime end, then you can take DGTP and you have your DNA strand, and then you add the enzyme terminal transferase. So this terminal transferase will transfer the guanosine residue onto the 3 prime group end of the DNA. Okay. So now how is homopolymer tailing used for synthesis of cDNA? As I was mentioning, that explaining that this mRNA will always have this poly A tail. Now, because it has this poly A tail, in the next step, you want to So because this mRNA has poly A tail, and you want to synthesize a cDNA from this mRNA, what is done is we take a oligo DT primer. Okay, an oligo DT primer is taken, reverse transcriptase, and the four different DNTPs: DATP, DGTP, DCTP, DTTP. And then what will happen? So once you take this reverse transcriptase, so it will use this primer, this oligo DT primer, and it will extend in the 5 prime to 3 prime direction, and you will get a new DNA strand. Now, now what is done? So you are getting a in the so what you are left with is you are left with a DNA RNA hybrid. Now you want to hydrolyze this RNA. Okay, so you want to hydrolyze this RNA. So what is done is this RNA DNA hybrid is passed into an alkaline sucrose gradient. So what this alkaline sucrose gradient will do, this alkaline environment will degrade the mRNA. Okay, the mRNA gets degraded, and you are left only with the cDNA. Now, using oligo DG and reverse transcriptase, what is that? This from this cDNA, again you get another DNA strand. Okay. So using this oligo DG, you synthesize a new strand of DNA. And then once this cDNA is obtained, you insert it into a vector, and then you grow the host cell. So basically, homopolymer tailing, what it is doing, it exploits the presence of poly A tail present on the mRNA to synthesize the first DNA strand, followed by degradation of the RNA template and synthesis of second DNA strand. So what are the steps? So in the first step, what we are doing, once we have the pure mRNA, we have isolated the pure mRNA. In the next step, we are using an oligo DT primer, and so we are setting up a reaction. So in the reaction, we are taking oligo DT primer. Along with it, what are we taking? We have to take an enzyme, so the reverse transcriptase enzyme and the DNTPs. Okay, nucleotide triphosphate. So this reverse transcriptase enzyme, it will bind to this oligo DT primer, and then it will, depending on the complementary, following the complementary rule, it will add the nucleotide, the complementary nucleotide one by one. Okay, now, so in the next step, what we will get? So in the next step, the synthesis of the, we will get the first strand synthesized, and then then in the next step terminal transferase is added. So in the next step the terminal transferase is added. So once the terminal transferase is added, you will get this 3 prime CCCC group. Okay, so to add C nucleotide, so you added DC oligo DCNTP, and in the 3 prime end you will get multiple cytosine residues added to the 3' end of both mRNA and newly synthesized first strand of DNA. So this C nucleotides will be added to both your newly synthesized cDNA and your mRNA as well. To the 3' end of the mRNA also this CC will be added. So now then you have do alkaline sucrose gradient. So this DNA RNA hybrid is loaded on an alkaline sucrose gradient. So when this is loaded on alkaline sucrose gradient, what will happen? This RNA will get hydrolyzed, and you will this will allow the recovery of the cDNA. This will allow the recovery of the cDNA. So this CCC will remain, and then using oligo DG primer, you have your 3' CCCC nucleotide, and using an oligo DG primer, you can synthesize the new DNA strand. Okay, using this cDNA as a template. So another method is the Gruber-Hoffman method. So here also, so here what is done? The same thing is being done here. So you are in both the methods, you are using an mRNA. First step is you have an mRNA, and so this is your template basically. So this is your template. So now for synthesizing a new DNA strand, what will be required? A primer will be required, isn't it? So since this mRNA has a poly A tail, an oligo DT primer is used, and then you have your primer, you will add your enzyme. You will add the reverse transcriptase enzyme. So from this mRNA, your cDNA will be synthesized. So once you get the cDNA, you are again getting an RNA DNA hybrid, isn't it? So this is your RNA strand, this is your DNA strand. You want to digest the RNA. So for digesting the RNA, you can use an enzyme RNase H. So what does RNase H do? RNase H is an enzyme which will specifically act on RNA DNA hybrid, and it will create breaks in this RNA molecule. So it will bind at the RNA DNA hybrid, and then it will create single stranded breaks in the RNA. And once these breaks are created, then you can add clinoenzyme. So once you add this clinoenzyme, then this breaks can be can the break that has been created, that can be again ligated. So it will polymerize, this clinoenzyme will polymerize this DNA strands, this broken DNA strands in the 5' to 3' direction, and new DNA strand will be produced. Okay, so basically in the two methods, so what is the difference between the first method and the second method? So in this homopolymer tailing, what you are using the alkaline sucrose gradient, alkaline sucrose gradient. You are using alkaline sucrose gradient so that you can degrade the RNA. Okay, and before loading this RNA DNA hybrid onto the alkaline sucrose gradient, you are first treating it with oligo DG, or you are treating with the terminal transferase, so that multiple cytosine base pairs can be added at the 3' end of the mRNA and the new DNA strand. Okay, and then you are synthesizing the new DNA strand using the oligo DG primer. Okay, and in the Gruber-Hoffman method, what you are doing the same thing. The only difference is that to degrade the RNA, you are treating it with RNase H. Okay. So these points are clear, right? So these are explained in the lectures as well. I'm just quickly brushing up your concepts. So answer this question. The following is used to bind mRNA specifically during the isolation of mRNA. So option A is poly A beads, poly C beads, poly T affinity column, poly G primer. So what is the answer? So your mRNA, what does your mRNA has? It has a poly A tail. So if you want to purify the mRNA from other RNA, what is required? You can answer in the chat as well. The answer is quality affinity column, isn't it? Answer is quality affinity column, because you know the DNA base pairing rule, right? The A will pair with T and G pairs with C. by 3 hydrogen bonds, A pairs with T by 2 hydrogen bonds. So this is basic molecular biology. So if you want to bind your mRNA, or if you want to particularly specifically purify the mRNA, then you need a resin. So this resin will have this oligo DT base pairs. And then once you pass this mRNA will have the poly A tail. So this poly A tail and this oligo DT resin so this oligo DT, the resin containing this oligo DT resin will bind to your mRNA, while all other RNA will get eluted. And then you can gradually elute this mRNA by treating it with particular concentration of NaOH. Yeah. So it is quality affinity column. So yeah, so this is the figure from the lecture slide. Okay, so you are first of all you are lysing. You have to lyse the cell. Then you will have or if it is a cell, you lyse the cell. If it is a tissue, you homogenize the tissue, and then so your lysate will have all the different types of RNA, right? All the RNA, DNA, and everything will be present. So you load this lysate into a column. So this column has beads, and this beads will have this oligo DT beads. So now you wash it with buffers to remove the non-specific binding, and then you elute it. Okay. So the poly-T affinity column isolate mRNA by binding specifically to poly A tails of mRNA molecules. So the poly-T affinity column, what will it do? It will bind to the poly A tail of mRNA, and the column contains oligo DT sequence that attach to these tails, allowing mRNA to be retained while other RNA species are washed away. The bound mRNA is then eluted from the column, resulting in purified mRNA. Okay. So I forgot that the method that exploits the presence of poly A tail on mRNA to synthesize first DNA strand. So actually, this question has some issue. Okay, because if you carefully understand both the methods, this Gober-Hoffman method and the homopolymer tailing, then in both the methods, what are you doing? So in both the methods, what is the first step? Your first step is you are having the mRNA and using the mRNA as a template. As a template, you are using an oligo DT primer. Okay, so you have your mRNA and your mRNA has poly A tail. Okay, poly A tail. Now, in both if you look at the steps in both the methods, the first step is that you are synthesizing the complementary DNA by using this oligo DT primer. So you are taking this oligo DT primer and then you are adding the enzyme and the DNTPs. So when you add the DNTPs, the primer and the enzyme, reverse transcriptase enzyme, only then your first strand of cDNA will be produced. Okay, so actually, the answer for this question should be both option A and D, because in both the methods, you are exploiting the presence of poly A tail on mRNA to synthesize the first DNA strand. Okay, but if you go and see the description that is there for this Gober-Hoffman and your homopolymer tailing, then in the homopolymer tailing, uh, the method that is there in your slide that mentions that. So this is the line that is there in in your lecture. In the lecture slide, it specifically mentioned that this method exploits the presence of poly A tail present on mRNA to synthesize first DNA strand. So they have given the answer as homopolymer tailing, but the answer should be both actually, because here also you are using this oligo DT primer for synthesis of the first DNA strand. Okay. So okay, the answer is homopolymer tailing, but okay. Now to produce nicks in the DNA hybrid, the following is used during the Gober-Hoffman method. So just now we mentioned, so if you so once you have synthesized the first strand of cDNA and you want to digest the RNA, which is present in your RNA DNA hybrid, then what enzyme is used? So it is RNase H. So this RNase H has the ability to specifically recognize the RNA DNA hybrid, and then on binding to this RNA DNA hybrid, it specifically digests, or it will cause nicks in this RNA strand. And once this nicks are created, then this enzyme is added, and then it what it does, it causes polymerization in the 5' to 3' direction, and it removes the these primers by 3' to 5' exonuclease activity. It removes the RNA primers. Okay. Or these RNA fragments that are there, basically. Answer is option C. So the RNase H is used to introduce nicks into the DNA RNA hybrids by specifically degrading the RNA strand while leaving the DNA intact. So your DNA will be left intact and your RNA strand will be degraded, and this cleavage creates breaks in this RNA, which is useful for subsequent analysis or manipulation of the DNA. So these points clear. So now this is again the same question, the type of primer used for the synthesis of first DNA strand in the homopolymer tailing method. So for the synthesis of first DNA strand, what type of primer will be required? Answer quickly. So your mRNA, so I've been repeating the same thing again and again. So your mRNA has this poly A tail. So what type of primer will be required? It will be, so A base pairs with T. So that is why oligo DT primer will be required. So for any DNA polymerase, any type of DNA polymerase uh, cannot cause de novo replication, which means that it will require a free 3' OH group. Only if a free 3' OH group is present, only then it can extend that 3' end of the DNA. So a primer is always required for DNA replication. So if you have studied basic molecular biology, you will know this. Okay, so that is why oligo DT primer has to be there. Only then your this reverse transcriptase and can come and extend the primer in the 5' to 3' direction using the DNTPs, the substrate and the complementary base pairing. The new DNA will be synthesized. Okay. So in homopolymer tailing method, oligo DT primer will bind to the poly A tail of mRNA, enabling the reverse transcriptase to synthesize the first strand of complementary DNA. The enzyme extends the primer by adding complementary nucleotides, resulting in a cDNA strand that matches the mRNA sequence. Okay, now what is this? So after, so what were the various steps? So first of all, the first step was isolation of gene. You isolated the gene, then, or the genome was isolated, then it was fragmented. Then you did then you created this recombinant plasmid, okay, by ligation. So you generated the recombinant plasmid by ligation reaction. So you ligated your vector with the vector with what? Vector with the gene of interest, and you got this recombinant plasmid, and then this recombinant plasmid is transferred to a host cell. Okay, it is transferred to a host cell. So this process in bacteria, this is called transformation. Okay, now your host cell. So there will be some bacterial cells. So let's take the example of bacteria, so that because it is most widely used and it is easy to understand. So some of your bacterial cell, what will it take? It will take one type of recombinant plasmid. Okay, so your genome will, once you have fragmented the genome, you will have different DNA fragments. So they will have different types of recombinant plasmid. So one type of recombinant plasmid will have, for example, gene X, one will have gene Y. So your these host cells will also have different type of recombinant plasmid. So now out of all these genes, you want particularly the X gene. So you have to or say you want a particular gene, you are interested in a particular gene, so you have to screen that which of this host cell has your gene of interest. So how is that done? So one method is by hybridization. Okay, so what is done in hybridization is basically you have a plate, you have an agar plate, say for example, and in this agar plate you have different colonies. So different type of colonies are growing on this agar plate. Now, what is done is first of all, a replica plate is prepared. So how a replica plate is prepared is basically once you, so uh, you can create an imprint of this in a separate agar plate. So you take a separate agar plate, and if you can label this, one method is you can label these colonies, and then you can transfer these colonies on a new agar plate. Or another method is you um, what to say? Yeah. So you plate this on a separate agar plate. Okay, new agar plate, and a replica plate is created. So your replica will plate will also have the same colonies. And now from this replica plate, these colonies are transferred on a nitrocellular. So you first of all you lyse the cells, denature the DNA, and bind to the matrix, and then these lyse cells are Lyses will be transferred to a nitrocellulose membrane, or it can be a PVDF membrane, or it can be Whatman paper. Any membrane you can transfer this on a matrix, and then because you have lysed these cells, it will have the DNA, and then you add a probe. So your probe is the will be complementary. The sequence of the probe will be complementary to your target sequence. Okay, and this probe will be labeled with a particular fluorescent tag, or it can be labeled with a radioactive molecule. Okay, so is it clear? So for example, so here in this figure, it has been shown that this particular colony has your gene of interest. So your probe will have sequence which is complementary to your gene of interest. So it will bind to this gene of interest, and it will give fluorescence, or it will give a particular radioactive signal. And now you can isolate this colony, and you can subculture it and grow it further. So you will have a homogeneous colony having this particular gene, your particular gene of interest. So all the colonies which are having this particular gene will be can be grown. Okay, so if you want to screen transform bacterial colonies with radio label probe by hybridization, that is your purpose. So now the colonies are gridded onto a master agar plate and onto a nitrocellulose or nylon filter. So this is your nylon filter. So the colonies that have grown on the filter are lysed in situ and processed for hybridization. So these colonies are lysed. So once you lyse the colonies, you will get the DNA. And now the probe will be used against the. So the probe can be anything. It can be a single-stranded DNA, or it can be an mRNA, which is complementary to the, or RNA can also be a single-stranded RNA as well. That is complementary to your DNA of interest. But it should have a radio level group or a fluorescent group, so that on binding to your target of interest, it gives a particular signal. Okay. So what are the steps? So first of all, the different steps in screening bacterial clones by hybridization is, first of all, you need to prepare a radioactive probe. So how do you prepare a radioactive probe, that we will learn. And then you need to prepare a replica plate. So once you prepare the replica plate, you need to blot it. So blot it on any membrane, a nitrocellulose membrane, for example, and then you hybridize it. So once you blot it, then you add the probe. Then you add the hybridization means what, you are adding the probe to your blotted to your blot basically, and then you develop the blot. You do autoradiography. If it is a radioactive probe, you can do autoradiography and detect the signal. Okay. So what are the methods of clone identification? So you see this figure nicely. So this figure is again taken from the book T.A. Brown. So what is done, the colonies are transferred to nitrocellulose or nylon membrane. So you see here, so this is your agar plate, which is having the bacterial colonies growing on it. Now you transfer these bacterial colonies to a nitrocellulose or nylon membrane. So you see, this is your nylon membrane, and now what you do, you uh, basically put the nitrocellulose membrane on your agar plate. So now your nitrocellulose membrane will also have the colonies growing on it. Okay. So the colonies will be transferred on the your nitrocellulose membrane. Now what you do? So in the next step, the colonies which have been transferred on this nitrocellulose membrane, they are lysed. They are lysed by treating it with alkali and protease. So now when you treat it with alkali and protease, you will get the free DNA. So the DNA will be extracted from the cell. Now for fixing this DNA, you want the DNA to be attached to your nitrocellulose membrane. So you treat it with ultraviolet radiation or you heat it at 80 degrees Celsius for 2 hours. So when you treat it, then your DNA will get attached to your nitrocellulose membrane. And so you see us after treatment, what you are getting, the DNA is getting bound by the sugar phosphate backbone. And then you are probing. In the next step, what you're doing in this step number 3, you are probing. So this is blot. So this is basically blotting. Blotting means you are hybridizing your DNA of it. You are hybridizing the DNA with the probe, which is having the labeled DNA. So you have different types of DNA which have been attached to your membrane. So you can see here after washing, only one particular probe is getting bound to your only one type of DNA is getting bound to your probe. And now you can detect this by using an X-ray film, and you can detect that signal is coming from only this particular clone. Okay, so the resulting is you are getting a positive hybridization. So I hope you understood the steps. So this all things are mentioned in the text as well. You can go through the text. The same thing is mentioned. So the colonies are placed that first transferred to a nitrocellulose or nylon membrane. So you see here they have been transferred on the nylon membrane. Then the membrane is treated. So you treat the membrane with alkali and protease. So once you treat with alkali protease, all the hydrogen bonds will be broken. So even your double-stranded DNA will get single-stranded. And now the membrane is heated for short period at 80 degrees Celsius. If a nitrocellulose membrane is being used, or with a nylon membrane, or if it is a nylon membrane, you can do UV irradiation. So the molecule now becomes attached to the membrane through their sugar phosphate backbone. Okay, which means that the bases are free to pair with complementary nucleic acid molecules. Now, in the next step, you add the probe. So it can be a single-stranded DNA which is having a fluorescent tag, or it is having any radio-labeled molecule. Now, this probe will bind to that DNA to which it is complementary, and then you can visualize it by autoradiography or by detecting the fluorescent signal. Okay. Okay. Now, how do you generate this? So your first step is the synthesis of the radioactive probes. So how do you synthesize radioactive probes? So what are probes basically? So probes are basically, it can be either single-stranded DNA to which you have radioactive molecule, or it can be uh, it is usually single-stranded DNA molecule. Okay, so single-stranded DNA molecule which is complementary to your DNA of interest. So one method is by nick translation. So what is done in nick translation? So you have your double-stranded DNA, and your double-stranded DNA is treated with DNase1. So once you treat it with DNase1, so there will be several single-stranded it will cause random breaks in your DNA strand. So in both the DNA strands, there will be random DNA breaks that will be caused by this DNase1. Now, once this DNase1 acts, then what is done? Then you treat it with clinoenzyme or DNA polymerase 1. So DNA polymerase 1 has another subunit which is called the clinoenzyme. So this clinoenzyme, and along with it, what do you use? You use 3 unlabeled DNTPs and 1 radio-labeled DNTP. So for example, you want the probe, your DNA probe to have radioactive adenine molecule. Okay, radioactive adenine molecule. Then you use DATP. So DATP having this P32, while other DNTPs will have normal phosphorus group, P31. So this enzyme, wherever it will, so it will use this it will consider this as one of the primer, and it will extend in the 5' to 3' direction, this DNA polymerase 1. And using the, so wherever there will be, for example, you have used a labeled DATP, then a complementary T will be added here. Okay, so in this way, you can get the labeled DNA probe. Okay, is it clear? So you see this figure, you will understand. Okay, this is not difficult to understand. You treat it with DNase one, you get several DNA, short DNA fragments. This multiple breaks are caused, and then you treat it with DNA polymerase one, and along with DNA polymerase one, you need to add one of the radioactive DNTP, and the other three DNTPs will be normal. So this whenever so that radio labeled DNTP that you are using, whenever it will have a complementary base pair, it will add that radioactive DNTP to that region. Okay, so you can read the text from here as well. Okay, now another method is random primer method. So what you are doing in this random primer method, so in the first step, you have your double-stranded DNA. So your double-stranded DNA is first denatured. So how will you denature the DNA? By heating it. So you will get two single strands. Two single stranded DNA will be separated, and now you add a primer. So once you add so it can be any random primer. So this random primer will bind to that part of the DNA to which it is complementary. So you see here random primers have attached to different regions of the DNA wherever the sequence is complementary to that region of the DNA. And then you add the cleano fragment and 4 DNTP. So once you add the cleano fragment and 4 DNTPs, what will happen? So again, in this 4 DNTPs, out of the 4 DNTPs, one will be radio labeled and another will be other three will be normal. Okay, so this will use this free 3' group and it will cause extension, and the depending on the radio labeled your radio labeled molecule will get attached when this cleano fragment start polymerizing in the 5' to 3' direction. Very clear. So that is random primer method. It depends on the ability of random hexanucleotide. So you are using hexanucleotide to anneal to multiple sites along the length of a DNA template. So hexanucleotide means any 6 nucleotide. It can be ATT, CG, or any random hexanucleotide is synthesized, and then the DNA is separated and this random primer is added to your DNA. So the primer template complexes will be formed, and this will be a substrate for the cleano fragment of DNA polymerase 1. Now, this enzyme synthesizes new DNA by incorporating nucleotide monophosphates at the free 3' OH group provided by the primer. So the primer is providing your free 3' OH group, and this free 3' OH group will be used by the cleano enzyme, and it will start polymerizing in the 5' to 3' direction. And out of the 4 the newly synthesized DNA is made radioactive by substituting a radio-labeled nucleotide or a non-radioactive one in the reaction mixture. Okay. So So if your ATP is radioactive, then it will then once it starts polymerizing, wherever it will find D in the template, it will add this radio labeled A to the new DNA strand that is getting synthesized. That is random primer method. Another is terminal transferase method. So in this terminal transferase method, what you are doing, you are first using, so you see this figure, you can go through the text here later. So this text is what was explained in the lecture as well. So you first treat it with lambda exonuclease. So exonuclease means what? That the DNA or the nucleic acid will be chewed from the end, from one end. So once it is chewed, what you will get this 3' end flanking, or uh, this 3' end. So what does this lambda exonuclease basically do? It cleaves the DNA from the to at the 5' end. The DNA which is present at the 5' end, that is chewed by this lambda exonuclease. It gets degraded. So now you are left with some sticky ends, or the ends of DNA, the 3' ends of DNA gets free. Now what happens is this terminal transfer is added. So I was explaining terminal transfer is in your homopolymer tailing as well. So here also the terminal transferase and radioactive ATP is used, DATP is used. So this what this terminal transferase does is it adds adenosine molecule. Adenosine molecule at the 3' end of the DNA. Okay, 3' end of this DNA, it will add adenosine terminal. So whatever, so what does this terminal transferase do is it basically whatever substrate you provide, whether you provide DATP or you provide DCTP or you provide GTP, depending on what you are providing, it will add that particular nucleotide at the 3' end. Okay. So it will label the probe at the ends to the last nucleotide of the probe. Last nucleotide of the probe. So the 3' is the last nucleotide. So it will add to the 3' on end. Always it will add at the 3' end. Okay. Now the preparation of a replica plate is important in screening methods too. So why is it necessary to prepare a replica plate? Because if you must have a stock basically, isn't it? Why do you create Xerox or copies? Because you do not want anything to happen to your primary source or your main copy should not get damaged. That is why you create replica of anything. So that is why if you have to carry experiments, so you are lysing the cells and then you are treating it with uh, uh, then you're treating it with the probe, with your radio label probe. So if you do this with your original stock, if you do all this treatment to your master plate, then what will happen? Then 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 you will not have any stock so for example, you are doing everything with the master plate. Then for now you have found out that okay, my colony 1 is having my particular gene of interest, my desired gene of interest. Then once you have identified your colony 1, then where will you get that colony 1? Because you have already degraded the colony 1, you have treated it with SDS and alkali and protease. So now you have destroyed your stock. So you do not have anything left to proceed your experiment or to work further. So that is why you are creating a replica plate. So that is the answer. Simple reason, you just read the option, you can answer. So to maintain the original genomic or cDNA library intact. Okay, you want to maintain the original copy of it. Okay, genomic or cDNA. Answer is A. You can go through the explanation. So it mentions the same thing. So it enables further use while performing specific screening on the replica plate. Okay. So the probe in DNA hybridization screening is used to. So why is it used? Probe, as we were mentioning, the probe is used to identify a DNA sequence by binding to a complementary sequence. So your probe will have this radio label group or a fluorescent group, and it will be complement to your DNA of interest. Okay. You can read this. This probe is single stranded and it is complementary to the target DNA. Okay. So once you add this probe, it will hybridize to your single stranded DNA. So you created the single stranded DNA by treating with alkali, heating it at 80 degrees Celsius. So I mentioned all the steps. So you go through those. So the answer is option B. Now the role of clinofragment in the random primer method of labeling a single stranded DNA probe is to. So what was the function of clinofragment? So clinofragment is basically a subunit of DNA polymerase 1. So what is your clinofragment doing? It is polymerizing in the 5' to 3' direction. So in the first step, what is happening? You are treating with the DNase 1. So you are creating this break. You are basically creating the free 3' OH group. Once you have the free 3' OH group, then your clinofragment can act. So if you are treating with RNase H, then also you are creating the free 3' OH group. And then this free 3' OH group can be polymerized in the 5' to 3' direction. So the answer is what? To synthesize a new DNA strand, because your clinofragment has 5' to 3' polymerase activity, so it can synthesize a new DNA strand. Okay. You can go through this description as well. Okay. By binding short random oligonucleotides to single stranded. Okay. Now there can be immunological method for screening as well. So one was the using of this probe, isn't it? Using this, one is the hybridization method. So in the hybridization method, you were using single stranded DNA probes for binding to your DNA of interest. Now, another method is the immunological method. In immunological method, you will use antibody against the your protein of interest. So you see here, see this figure. So this is the membrane on which your colonies are growing, and you digest the or you treat the colonies or lyse the cells, and then once you lyse the cells with chloroform, you will have different proteins which will be present inside the cells. And now you can use antibody against this particular protein. Okay, so if the particular colony is expressing your desired protein of interest, or it is having the particular desired gene of interest, then also it will express the particular protein, your desired protein. So you can use antibody against this particular protein, and then you can analyze the signal. So you get this antibody, and this is your primary antibody. Then you add a secondary antibody. So this secondary antibody will be specific to your primary antibody, and the secondary antibody will be conjugated to a fluorophore. Okay, so that you can enhance the signal basically. So several secondary antibody molecules can bind to a single primary antibody molecule, increasing the amount of signal. So what is the purpose of using a secondary antibody? Because is the only reason is that it is increasing the amount of signal that is produced and enabling a clearer detection of each positive colony. Okay. Are these points clear? These points are clear, right? Immunology will lyse the cells, you will have the particular protein present in your cell, then you will use antibody against your protein of interest. So only those colonies which are expressing your protein of interest will bind to your antibody, and then you can use secondary antibody which will be specific to your primary antibody. And this secondary antibody, because it is conjugated to fluorophore, it will enhance the signal and allow for easy detection. Now, the purpose of using labeled secondary antibody in immunological screening, what is the reason? Just now I explained that it is the only reason is to enhance the signal for detection. Answer is option D. Okay, to enhance the signal for detection. So amplifying the signal binds to multiple site on primary antibody. Now coming to polymerase chain reaction. So if the target sequence is known, if you know if you want a particular clone a particular gene of interest. So, for example, you want to work on the protein, say, for example, hemoglobin. You want to study about the hemoglobin protein which is present in, say, humans, hemoglobin protein of human. Then you will know the gene, the the gene which codes for hemoglobin, that is already known. The sequence of the human gene which codes for hemoglobin is already present in your database, in our genome database. NCBI database will have all the sequence for particular protein. Now you know the particular gene, you know its sequence, and then you can directly do polymerase chain reaction and amplify your gene of interest, and then clone it in a suitable vector and express it in a host cell. Okay, so we are all about this polymerase chain reaction. So in polymerase chain reaction, what is done? It is a selective amplification of a chosen region of DNA molecule. So if you want to amplify a particular region of DNA, then you go for PCR reaction. So in this, what is done? Any region of DNA molecule can be chosen as long as the sequences at the borders of the region are known. So it is necessary that you know the sequence of the gene which you want to amplify. If you want to perform polymerase chain reaction, then it is necessary to know the sequence of the gene, because based on that you will design your forward primer and reverse primer. So there are three steps. In the first step, there is denaturation. Then so in the denaturation, the two strands of DNA get separated. Then there is annealing. So in the annealing, what happens? Your forward primer and reverse primer will align to your the ends of the gene, and then extension. So yeah, Taq polymerase will extend this primer. And so this cycle, you can repeat this cycle and you can amplify your gene of interest. So the border sequences must be known. Two short oligonucleotides. Why? Because two short oligonucleotides are used to hybridize to the DNA molecule, one to each strand of the double helix. These oligonucleotides, which act as primers for the DNA synthesis reaction, delimit the region that will be amplified. So basically, polymerase chain reaction, what we are trying to do, we are trying to we are trying to mimic the or we are trying to do in vitro replication also. In a way, we are doing in vitro replication. So replication is taking place inside our cell in the S phase of the cell cycle. Our DNA gets duplicated. Now, if we, in a similar manner, if we want to amplify a particular region of DNA, then we can do it in vitro by using what are the three things that we will be requiring. What are the major things that will be required for poly? First of all, you will require a template. So template DNA is the DNA is that DNA whose gene you want to amplify. So you will require a template DNA. Template DNA will have your gene of interest. Okay, so you will require a template DNA. Next, what will be required? You will require primers, a forward primer and a reverse primer. Okay, because your DNA polymerase, none of the DNA polymerase can start synthesizing DNA on their own. They require a primer for extending the causing DNA replication. Okay. Next, what will be required? You will require an enzyme, a polymerase enzyme. Okay. You will require a polymerase enzyme. And next, what is required? We will require DNTPs. Anything else? Is it required? You will require buffer. So for your enzyme to act, you will require a particular buffer which will have different cofactors like magnesium chloride, so that for the activity of the enzyme. So these are the five different things that will be required for doing a polymerase chain reaction: template DNA, primers, polymerase, GNTPs, and buffer. Okay. Now, what are the steps in a buffer in a PCR reaction? So there are three steps: denaturation, annealing, and extension. Okay. So during each cycle of PCR reaction, what will happen? In the first step, denaturation, your DNA will get separated. You will get single-stranded DNA, and then your primer can anneal to your DNA, single-stranded DNA, and then so DNA, what is the temperature during denaturation step? 94 degrees Celsius, which will break the base pairs. So it will break the hydrogen bonds. Then hybridization or annealing temperature, at which what will happen? Then the temperature is reduced. So from 94 degrees Celsius, the temperature is reduced to what temperature? It is around 58 or 55 to 65 degrees Celsius. This is the annealing temperature. So in this annealing temperature, what will happen? The primers will anneal to the single-stranded DNA. And then the extension temperature, at with this temperature, what will happen? The polymerization reaction will take place. At which what will happen? This Taq polymerase will come and it will extend the DNA. Okay, it will bind to the primer and it will extend in the 5' to 3' direction. Okay, so what are the essential components? You require a thermostable DNA polymerase, which is Taq polymerase mostly. Then you require a primer, DNTPs. You require buffer to maintain the pH. Buffer will also have this cofactor divalent cations, mainly magnesium plus, magnesium 2 plus, and you require template DNA. Okay, so what are the steps one by one if we see? So in the denaturation, what happens? So the denaturation temperature is usually kept 94 degrees Celsius, but your denaturation template will depend on your, what will it depend on? It will depend on the gene sequence or the sequence of gene that you want to amplify, your target gene. What is the sequence of your target gene? So if it is rich in GC, if it is rich in proportion of the GC is very high, then what will you require? You will require a higher denaturation temperature, because there are triple bonds between guanine residues and cytosine residues, so you will require a higher denaturation temperature. So the temperature will depend on the GC content, and also it will depend on the length of DNA molecule. If you want to amplify a target DNA sequence which is very large, then the denaturation temperature has to be increased, as well as the denaturation time has to be increased as well. Okay, so if the temperature is low or time is too short, so you have to optimize it, that what should be the temperature, what should be the time. Then what will happen if you do not, if you keep it very high, if the temperature is kept low, or if the time is kept very short, then what will happen? The entire DNA region, the target DNA will not get separated. Okay, only 80 rich regions of the template DNA will be denatured. Okay, For example, you have performed a PCR reaction and then you are not getting any band when you are running it on a gel. So these are the points that you have to keep in mind, that what is my DNA sequence, what is the length of my DNA. So all these have to be kept in mind. So in PCRs catalyzed by tagged DNA polymerase, denaturation is usually carried out at 94 to 95 degrees Celsius, which is the highest temperature that the enzyme can endure for 30 or more cycles without sustaining excessive damage. So this stack polymerase is a thermostable enzyme. So it can cause, so it does not get denatured at even 94, 95 degrees Celsius. Okay. So the denaturation is usually carried out for 5 minutes. So the initial denaturation, this is called initial denaturation, to increase the probability that long molecules of template DNA are fully denatured. So you have a initial denaturation which will take place in only once. This initial denaturation takes place only once, and then you can create the cycle. So at which step denaturation, annealing, extension, and this cycle you can set how many cycles you want. Okay. Now next is annealing of primers. So the primers, what are the different things you have to keep in mind? The temperature that is to be used for annealing. Annealing temperature is lower than the has to be lowered. So denaturation temperature are higher and the annealing temperature is lower. Now, if the annealing temperature is too high, if it is too high, then the primers will not anneal to the template and yield of DNA will be very low, right? Now, if the TA is too low, if the TA, so there are a couple of points you have to keep in mind. So if the TA is too low, then what will happen? Your separated DNA strands will start to anneal. Okay, there will be non-specific annealing of primers, resulting in amplification of unwanted segments of DNA. So if you keep the annealing temperature low, then this primer can bind anywhere on the DNA. That is also one problem. And it must be low enough to enable hybridization, but and it must not be very high. If it is very high, then it will not anneal. If it is very low, then it can show non-specific binding. Okay, so this temperature can be estimated by determining the melting temperature or the TM of the primer. So when you design a primer, you will have a melting temperature. So you can determine the melting temperature of the primer. So what is melting temperature of primer? So melting temperature is that temperature at which 50% of your primers will anneal to your DNA molecule and 50% will not anneal to your DNA molecule. Okay, so the annealing temperature has to be lower than the. So you can see this figure. If it is too low, it is going and binding anywhere on the DNA. Okay. So this will give you mismatched hybrid and not correct result. So you have to set a correct annealing temperature, and this step requires a lot of optimization. You may have to run several PCR reactions and see at what temperature. So you have to do a gradient PCR and see what is that particular temperature at which your PCR primer is getting bound to DNA. Okay. So you have to keep these points in mind, and your annealing temperature must be lower than the melting temperature. Okay, so your melting temperature must be greater than your annealing temperature. Okay. So every primer has a particular TM. You can determine the TM of your primer. Okay. The TM is the temperature at which correctly base-paired hybrid dissociates, melts. So at the temperature, so usually the annealing temperature is usually kept 1 to 2 degrees Celsius below this, because this should allow enough. This temperature should be enough to allow correct primer template hybrid to form, but too high for a hybrid with a single mismatch to be stable. Okay. The two primers should be designed so that they have identical melting temperature. Okay, so the two primers that you design, the forward primer and the reverse primer, these are their annealing temperature must not be very different. They must have identical. There can be 1 or 2 degrees Celsius difference, but it must not vary greatly. Okay. So the temperature, if this is not the case, appropriate for one primer may be too high, one might be too low, and this will create improper annealing. Then in the extension, what has happened? It is carried out at 70 to 78 degrees Celsius. Now, the time of extension that has to be optimized depending on what type of polymerase enzyme you are using, whether you are using a Taq polymerase or you are using a fusion polymerase. So different polymerases have a polymerization rate. So Taq polymerase has 2000 nucleotides per minute. This is the polymerization rate for Taq polymerase, which means that 1 minute around polymerization rate 2000 nucleotides per minute, and extension is carried out for 1 minute for every 100 base pair of the product. Okay. So depending on what is the length of your DNA that you want to amplify, you can set that how many minutes you have to keep. because your polymerization rate is 2000 nucleotides per minute. And if you want to amplify, for example, 4 kb DNA or 10 kb DNA, then accordingly you have to set the minute. Okay. So the number of cycles will depend on the number of copies you want to generate. And at least 25 cycles are required to achieve acceptable levels of amplification of target sequence in mammalian DNA templates. Okay. Now, what is the correct event of main events order? What is the correct order of main events involved in polymerase chain reaction? So, what is the order? First is denaturation, followed by annealing, and then extension. Isn't it? Extension. So, answer is what? Denaturation, annealing, and extension. Now, the way the number of DNA molecule increases during PCR cycle. So what happens? You have your DNA molecule. So from one DNA molecule, what happens? This DNA molecule is first separating into two separate DNA strands, and you are getting a new DNA strand from this separated DNA strand. So from one, you are getting two. Then from two, what? How many you will get? You will get four. Okay. Then you will get what? Eight. Then you will get 16. So this is exponentially increasing. So it is 2 to the power 1, then square 2 cube, 2 to the power 4, then 2 to the power 5. Okay. So this is exponentially increasing. So answer is exponential. So any exponential bacterial growth curve, anything, it goes by this formula. N equal to N naught into 2 to the power N, where N is the number of cycles and N naught is the initial number of DNA molecules, say for example. Okay, so for bacterial growth curve also, this was the formula. Any exponentially growing or exponential reaction follows this formula. Okay, initial DNA concentration. So it is exponential. There is another equation also. So you can use this formula: C equal to C naught 1 plus E to the power N, where E is the efficiency of PCR amplification. Okay, number of cycles, slope of exponential phase, also this, but this is the simple formula. Uh, okay. Now, Taq polymerase is the enzyme. So, what is done is this Taq polymerase is usually used for if you want to screen or if you want to identify whether your clone has your gene of interest. Okay, then you can use Taq polymerase. While if you want to do any site-directed mutagenesis, then we usually do not use the Taq polymerase because it is very error prone. Okay, the enzyme is very error prone and it lacks proofreading activity. So any enzyme, if you have read about the three polymerases enzyme, DNA polymerase 1, 2, 3, and this replication process, then you will be knowing that these polymerase enzyme has a proofreading activity. So this proofreading activity of any enzyme is conferred by which property? Proofreading activity by the 3' to 5' exonuclease activity. So if any enzyme has this 3' to 5' exonuclease activity, then it is called a proofreading activity of enzyme. So the polymerase enzyme synthesizes in the 5' to 3' direction, right? It synthesizes in the 5' to 3' direction, and what it does, it proofreads in the 3' to 5' direction. So, for example, you are writing an essay, for example. So what you do, you write a line and then you go back and check that whether you have made any mistake. So in a similar way, your polymerase also works in the same way. It is synthesizes in the 5' to 3' direction, and then if that enzyme has 3' to 5' exonuclease activity, then it will go back and then it will check whether it has made any mistake. So if it has made any mistake, then it will use this 3' to 5' exonuclease activity. So what does 3' to 5' exonuclease activity means? It means that it will degrade the DNA. If it has made any error, then it will degrade or it will replace that particular nucleotide with the correct nucleotide. So using the exonuclease activity, it will degrade that or it will destroy that particular nucleotide and it will add a new nucleotide, the correct nucleotide. Okay. But this proofreading activity, this 3' to 5' exonuclease activity is lacking. So the Taq polymerase lacks this proofreading activity. Okay, that is why it creates several errors. Taq polymerase does not have 3' to 5' exonuclease activity. Remember this. Okay. Okay, so the tag polymerase lacks some proofreading activity and therefore it is unable to correct its errors. This means that the DNA synthesized by tag polymerase is not always an accurate copy of the template molecule, and the error rate is approximately one mistake for every 9,000 nucleotides. So for every 9,000 nucleotide it synthesizes, there will be one error usually. Okay, however, this translates to one error in every 300 base pair for PCR products obtained after 30 cycles. So if it is making one error in 9,000 in every 9,000 nucleotides, then this error will get multiplied. So because it is an exponential growth, it is following exponential growth, the error will also get replicated. So at the end of 30 cycles, there will be one error in every 300 base pairs. Okay. So what distinguishes tag polymerase from other polymerases? So it lacks 5' to 3' exonuclease activity. This is wrong. It lacks 3' to 5' exonuclease activity. It lacks the proofreading activity. Okay, the proofreading activity. And the 3' to 5', 5' to 3' exonuclease activity is required for digesting the RNA primers. Okay, 5' to 3' exonuclease activity is required for digesting the RNA primers, and this 3' to 5' is required for proofreading. Okay, so answer is C. Okay. Now the primers used in DNA process in the PCR processes are what? So what type of primer do we use? So they are they are DNA primers only, but they are single stranded. So they are single stranded DNA oligonucleotide. The forward primer and reverse primer, they are both single stranded DNA oligonucleotide. Okay. What is the role of magnesium chloride in PCR reaction? So the magnesium chloride is basically in your buffer the PCR reaction you set up, there is a you have to add buffer. So this buffer contains the magnesium chloride. So magnesium is basically a cofactor for the Taq polymerase enzyme. So this Taq polymerase enzyme, so certain enzymes require cofactors for their activity. So for its activity it requires magnesium. So in the biochemical reactions that I was discussing in the previous live session, we discussed that there are several cofactors which are required, like calcium ions or these ions. So this Taq polymerase requires this magnesium. So answer is it is a cofactor for the Taq DNA polymerase. Okay, answer is option B. Now PCR has several applications. PCR was used for this in COVID as well. So to identify whether a person has a COVID infection or not, then also PCR can be used because from the sample you can isolate the RNA of the sample and then you can do a real-time PCR as well. Okay, and it can also be used for parental testing. So from the sample, you take the DNA sample from parent, you take the DNA sample from the child, and then there are particular DNA regions which which do not show any recombination. So, and there are several copies of it. So, depending on what is the number of copy, what is the pattern of DNA fragment, you can identify that whether this particular child is of this parent or not. Okay, so matching DNA patterns with crime scene evidence, detecting disease predisposal, characterizing the DNA markers in the child, mother, and father. So there are specific DNA markers which are transferred as it is from the parent to the offspring. There are certain chromosomes where which has particular DNA repeats, random repeats of DNA. So you must have read this in DNA fingerprinting. So these particular regions, if you compare between the DNA of the mother, father, and the child, then you can identify um, identify or you can this is done what is done in parental testing. Okay. So there are specific DNA markers which can be identified from child, mother, and father by comparing these markers which are inherited from the parents, and it can be determined if the child is biologically related to both parents. Okay. Now this is one of the formula for determining the melting temperature of a primer. You use this formula. What is this formula? This is 2A plus T plus 4G plus C. This is the formula that is used. Answer is option A. Okay, not B. Option A, 2A plus T plus 4G plus C. Estimates the melting temperature of DNA primers by calculating the number of bonds between hydrogen bonds between 80 base pair. Say 80 base pair has 2 bonds each, and GC base pair contribute to 4 bonds. So this formula helps predict the temperature at which half of the DNA strands are double stranded and half are single stranded, which is important for optimizing PCR reaction. So that is all. I hope all these questions were useful. Okay. Okay. So I hope all these concepts of this gene cloning and PCR reaction and the screening genomic library, cDNA library, was clear. And now you can attempt the assignment questions. And if you have any doubt in the assignment question and any particular section which you want to discuss, then we can take those sections in the next class as well. So I hope this session was useful. So thank you, everyone.